In this lecture, we will be looking at the next class of biological molecules, referred to as lipids. Lipids are a diverse group of hydrophobic molecules. I want to emphasize the word diverse. There are many different types of lipids, and they have many different structures. Lipids are the one class of large biological molecules that do not form polymers. In other words, they do not consist of many hundreds or thousands of similar repeating units. They are quite different, actually. The unifying feature of all the diverse types of lipids is that they all have little or no affinity for water. In other words, lipids are hydrophobic, either completely hydrophobic or partially hydrophobic. They consist mostly of hydrocarbons. This means many hydrogens and carbons attached to each other, which form nonpolar covalent bonds. Because lipids are hydrophobic, they mix poorly or not at all with water. The most biologically important lipids are fats, phospholipids, and steroids. Fats are also called triglycerides. Or triacylglycerols. Fats are important for mammals, such as ourselves, for energy storage. Phospholipids are the major components of all cell membranes. Steroids are important for membrane structure as well, in particular cholesterol. Also, many vertebrate sex hormones are steroids. These are certainly not all examples of lipids. There are many others as well. Waxes are lipids. Some pigments are lipids. Any biological molecule that is at least partially hydrophobic would be put in this category called lipids. We will begin by discussing fats, also known as triglycerides or triacylglycerols. Whereas your book refers to them as triacylglycerols, most of the time you will hear the term triglyceride, and the term triglyceride is also used in your lab manual. Be sure that you understand that these are in fact the same thing. Every fat molecule is constructed from two types of smaller molecules that include one unit of glycerol and three fatty acids. Shown above is glycerol. Glycerol is an alcohol. Notice that it has three hydroxyl groups, which are common in alcohols. At the top right of the screen, you're looking at one fatty acid. Notice that the fatty acid has an important chemical group that I'm circling here. This group, this COOH group, is a carboxyl group. Attached to the carboxyl group is a chain of hydrocarbons, hydrogens and carbons linked together. And it's this region of the fatty acid, the hydrocarbon chain, that is nonpolar and makes fats hydrophobic or water-fearing. In order to build one molecule of fat, or one triglyceride, we must take three fatty acids and join them to this glycerol. That happens with three consecutive dehydration synthesis reactions, the first of which is illustrated above, where the hydroxyl group of glycerol and the carboxyl group of the fatty acid are going to interact, and a very specific enzyme will remove two hydrogens and an oxygen, and in their place a new covalent bond will form. This type of covalent bond is referred to as an ester linkage, and there will be three of them once we are done building our single molecule of fat. So this bottom illustration is one fat molecule, or one triacylglycerol or triglyceride, which consists of one glycerol linked to three fatty acids, each of which is connected with an ester linkage. and results from dehydration synthesis. This particular molecule of fat that is shown is what we would call a saturated fat because it has the maximum number of hydrogens attached to every fatty acid. Notice that all the carbons are joined with single bonds. That means that each carbon can be attached to 2 hydrogens or 3 if it's the terminal carbon. So the maximum number of hydrogens are attached, and this fat is saturated with hydrogens. The major function of fat is in energy storage. So humans and other mammals store energy in 2 very different types of biological molecules. Recall from the previous lecture that we can store energy in our liver and muscle cells in the polysaccharide known as glycogen. Polysaccharides are very different from fats. Fats can also function in energy storage, but this is done in a different type of tissue, referred to as adipose tissue. So fatty tissue is energy storage tissue called adipose. 1 gram of fat will store over twice the energy as 1 gram of polysaccharide. So fat is a more efficient way to store energy. The fatty acids that are within a single molecule of fat can vary in a couple ways. They may vary in the number of carbons on the fatty acid. Typically, there are about 16 to 18 carbons on a single fatty acid. And they can also vary in the number and locations of double bonds between the carbon atoms of the fatty acids. As I mentioned on the previous slide, we were looking at an example of a saturated fat. Saturated fats have the maximum number of hydrogen atoms attached to their hydrocarbon chains. There are no double bonds between carbon, therefore maximum numbers of hydrogens are attached. And this is what we see when we look at most animal fats. Saturated fats are solid at room temperature. And an example of that is shown above as we look at this cube of butter, which is a saturated fat. If we zoom in on the molecules that make up this butter, here is one of the triglycerides shown. Again, 1 glycerol linked to 3 fatty acids. And each of these fatty acids has the maximum number of hydrogens attached. There are no double bonds at all between carbons. It is saturated and solid at room temperature. Not all fats are like this, however. Some fats, such as olive oil, are liquid at room temperature, and we say they are unsaturated. What this means is the fatty acids have 1 or more double bonds in their hydrocarbon chains. Therefore, fewer than the maximum number of hydrogens are attached, and this gives the fats different properties. For example, they are liquid at room temperature. As you look at this illustration to the right, an unsaturated fatty acid is shown at the base. There's a double bond between these 2 carbons, and that means there will be fewer hydrogens attached, and this particular fat will be liquid at room temperature. The illustrations shown here are shorthand. Notice that the fatty acids don't show any symbols for carbon or hydrogen, so you have to understand what this shorthand representation means. Every point represents a carbon, and every line is a covalent bond between the carbons. Hydrogen atoms are simply inferred. So let's go ahead and draw this on the next slide to illustrate a little bit more clearly. Let's begin by illustrating one molecule of glycerol. Glycerol has 3 carbons, which are attached to 3 hydroxyl groups. Recall from previous lectures that every carbon atom will form 4 bonds with other atoms, so we have to fill in all the gaps with hydrogens to satisfy this rule. Now we'll go ahead and draw our fatty acids. We will need 3 of them to build 1 molecule of fat, or 1 triglyceride. Each fatty acid has a carboxyl group, which is a carbon double bonded to an oxygen, single bonded to another oxygen, which is attached to a hydrogen. There's your COOH, that's your carboxyl chemical group or functional group. And then attached to that carboxyl group is the hydrocarbon chain of the fatty acid, which is typically 16 to 18 carbons long, but for simplicity, we will draw this one much shorter. 1, 2, 3, 4, 5 carbons long. Since every carbon is going to form 4 bonds with other atoms, we need to fill in the gaps with hydrogens. So this first carbon already has 2 bonds with other carbons. It needs 2 more. We'll fill those in with hydrogens. Same with this carbon. And my pen's getting a little crazy here. And this carbon. And then the last carbon atom only has 1 bond with its adjacent carbon, so it needs 3 more bonds, which will all be hydrogens. This right here is a saturated fatty acid. All the carbons are single bonded to one another, therefore you have the maximum number of hydrogens attached. If I were to draw this same exact fatty acid in shorthand notation, it would look a little different. First of all, I will do the carboxyl group. But for this hydrocarbon chain, I need 1, 2, 3, 4, 5 points to represent my 5 carbons. 1, 2, 3, 4, 5. So every point is a carbon. And the hydrogen atoms are simply inferred. So if we go back to that previous illustration, as you look at these pictures of fatty acids, every point is a carbon and hydrogens are inferred. For our third fatty acid, let's go ahead and draw one that is unsaturated. First, we'll start with the carboxyl. And then our chain of carbons. But let's go ahead and put a double bond in here. Let's say that these 2 carbons are connected to one another with a double bond, instead of the single bonds we have drawn previously. Considering the fact that every carbon forms 4 bonds with other atoms, this carbon down here that I'm highlighting needs 1 more bond. It has a double bond here, so that's 1, 2 covalent bonds. Here's the third. It needs 1 more, and we'll fill in the gap with a hydrogen. The carbon that it is double bonded to on the left side also has 3 bonds currently, and it needs 1 more. So we'll give it another hydrogen. And then you should be able to fill in all the gaps with hydrogens. The carbon at the very end needs 3 to fulfill its 4 bonding rule. My pen just got crazy again. This carbon needs only 2 more. And this one needs 2 more. Again, the pen is going crazy, but you get the idea. This last fatty acid that I drew is unsaturated because of this double bond between carbons. That double bond results in fewer hydrogens being attached as seen with all single bonds. Therefore, this fatty acid is unsaturated, and the more of these double bonds we introduce, the more unsaturated your fat will become and the more liquid it gets at room temperature. Now, if we're going to build a molecule of fat, we have to attach these 3 fatty acids to our molecule of glycerol on the left. And that will take place by 3 dehydration synthesis reactions. The hydroxyl group of glycerol will interact with the carboxyl group of a fatty acid, and these atoms will be lost. In their place, a new covalent bond will form, and we've attached our first fatty acid. That happens again here, and we lose another 3 atoms. And again here, and we lose another 3 atoms. So, in the process of building a fat, we lose 3 molecules of water, and we produce 3 new covalent bonds to hold our fatty acids to our glycerol. And then finally we have a molecule of fat. Another type of lipid, and actually the most important lipid for you to know in this class, is the phospholipid. Phospholipids are the major components of all membranes. So far, you've only heard of one type of membrane, and that is the cell membrane, or the plasma membrane. But eukaryotes have many other membranes inside of their cells, and phospholipids are the major component of those intracellular membranes as well. Phospholipid structure looks similar to that of a fat molecule. There is one molecule of glycerol, and to that we attach fatty acids, but this time we only attach 2 fatty acids, not 3. In place of the third fatty acid is the new group, which is a phosphate group. A phosphate group is a chemical group with 1 phosphorus atom and 4 oxygen atoms, and phosphate groups always have negative charges. We're going to simplify our illustration of a phospholipid like this. So our glycerol is here. We're going to link 2 fatty acids to it, and then at the top we're going to have our phosphate, our charged region. So that altogether would be our phospholipid, the charged phosphate head connected to a glycerol, and then 2 fatty acid tails. The 2 fatty acid tails are hydrophobic, but the phosphate group at the head of the phospholipid is charged, so it is hydrophilic. When phospholipids are added to water, they self-assemble into a bilayer. This means 2 layers, with the hydrophobic tails pointing toward the interior and the hydrophilic phosphate heads pointing to the exterior, where they interact with water. The structure of the phospholipid bilayer arrangement is found in cell membranes. Here's a closer look at our phospholipid structure. On the left-hand side is a more detailed structural formula. In the middle, you can see glycerol. Notice the 3 carbons of glycerol, and there used to be 3 hydroxyl groups here too, but because dehydration synthesis has already happened, these hydroxyl groups were joined with the carboxyls of 2 fatty acids, so we have 2 ester linkages here, and then the other hydroxyl group reacted with a phosphate, so we have another covalent bond up here. So we have 1 glycerol, we have 2 fatty acids, which are hydrophobic, and then at the top we have a phosphate group with some other atoms, and there's always a charge up here, and so the head of a phospholipid is hydrophilic. Recall that anything with a charge or a partial charge is hydrophilic and will interact with water. The space-filling model of a phospholipid is shown in the center, and then to the right we have our simplified version, which we will use in this class. Because phospholipids are part hydrophilic and part hydrophobic, we say that they are amphiphilic, or sometimes amphipathic. Amphiphilic. Part of the molecule interacts with water, and part of the molecule repels water, or is repelled by water. Because of this amphiphilic nature, we see the phospholipid bilayer forming in cell membranes. If we take a cell and we slice it in half so that we can see the layers. And then zoom in on this outermost region, this layer here. Then we can see what we're looking at on the picture. Notice that we have an upper layer of phospholipids and a lower layer of phospholipids. So right here is a phospholipid, and right here is a phospholipid. We have a bilayer of phospholipids that make up the membrane. There is always water outside the cell, and there is always water inside the cell, in the cytoplasm, and that forces this arrangement of phospholipids so that the phosphate heads are interacting with water, both inside and outside the cell, and their fatty acids are pushed to the center where there's no water at all. So the interior of this membrane has no water in it. That's the only way the fatty acids can arrange like this. There's another important lipid that we see in animal cell membranes, such as ours, and that will be embedded periodically in this bilayer. So I'm just drawing in a little structure to indicate to you a third class of lipid that we will find within a membrane, and that right there is cholesterol. Which brings us to a third category of lipid, steroids. Steroids are lipids that are characterized by a very different structure than you saw previously. Steroids have a carbon skeleton with 4 fused rings, and you can see that in the picture below. There are 4 fused rings. Everywhere you see a point, there's an inferred carbon, and hydrogens will be attached to that as well if there's room for a third and a fourth bond. Cholesterol is shown below here. Cholesterol is very important in our cell membranes because we are animals. Other organisms, like fungi, use a different type of steroid in their membranes, called ergosterol, but it looks very similar and it does the same thing for the cell membranes. In the case of cholesterol in our cells, it is very important for the cell membrane fluidity and integrity. It is a rigid structure that prevents gelling of the membrane at decreased temperatures and also prevents melting of the membrane at increased temperatures. So the membrane can maintain this semi-fluid, flexible nature, which is essential for life's processes because some things have to cross the membrane. Some things have to get into the cell or move out of the cell, and you have to have some sort of flexibility in the membrane to allow for that transport. Cholesterol is made in the liver, and it is shuttled to membranes where it is embedded in that phospholipid bilayer. Although cholesterol is essential in animals, high levels in the blood may be dangerous and contribute to cardiovascular disease. Not only is cholesterol important for cell membranes, but it is also important because it is a precursor for other steroids. Some vertebrate sex hormones are steroids that develop from cholesterol precursor molecules.